Okay, so it's 4 minutes past the hour, so I think we can get started. So good afternoon, everyone. Good morning or good evening, wherever you are. Welcome to the Palaeoverse Lecture Series. This lecture series is focused on software development and methodological approaches in paleontology, with the aim of promoting the use of new tools in the field and to give researchers a platform to showcase their work. With this series, we hope to shake up the usual type of lecture by having a mixed format with time dedicated to the presentation, demonstration, and discussion of the software or method presented. Now, typically, these sessions will run for about 50 minutes in total, and we will run this series throughout the year on the last Thursday of each month. We have one more lecture in the summer series coming up, and we'll announce the next lineup of speakers soon. And if you're interested in presenting a new method or software you have been developing or implemented, please feel free to reach out. So before moving on, just a few quick introductions. My name is Lewis Jones, and with me today I have Erin Dillon and Bethany Allen, and the three of us are on the development team of Palaeoverse, an organization aiming to bring the paleobiology community together, share resources, reach agreed standards, and improve reproducibility in paleobiological research. And if you want to know more about Palaeoverse, please check out our GitHub organization and website, and I believe those links will be dropped into the chat now. But that's all from me for now. Bethany will now give you some helpful tips and instructions for this webinar, followed by Erin, who will introduce this month's speaker. So thank you very much, and please enjoy this lecture. Over to you, Bethany.
Thank you, Lewis. So as Lewis mentioned, the format of the seminar will consist of a talk by our speaker, followed by 10 to 15 minutes at the end dedicated to questions and answers and discussion. My fellow hosts and I will help field your questions and moderate this discussion. Please note that this seminar will be recorded and will be made available on the Palaeoverse YouTube channel. However, we'll stop the recording before the question and answer period in case you prefer not to be recorded. Before we get started, I'd like to quickly point out a couple of useful Zoom controls at the bottom of your screen. So there's a question and answer button. You can use this to submit questions to our speaker. During the Q&A session, we'll either read out your question or invite you to turn on the microphone if you'd like to ask the speaker directly. There's a raise hand button, which you can use to raise your hand, and we'll call on you and invite you to turn on your microphone so you can ask the question. There's a chat button. Currently, you can only chat with the host in case of technical difficulties, but we'll allow you to chat with other attendees during the Q&A period. And also closed captions, and you can turn that on and view the transcript at any time. Lastly, I'd like to highlight a couple of ground rules. Our aim is to cultivate an open, respectful, and inclusive environment during this lecture series, where we can learn from each other. With this in mind, we ask that you please be respectful and kind when engaging with the speaker and fellow attendees, particularly when asking questions and interacting with others in the chat. Any form of harassment or inappropriate behavior will be grounds for removal from the webinar platform. For more information, please refer to the Palaeoverse Code of Conduct, which can be found on our website, and there'll also be a link in the chat. And with that, I'll pass it over to Erin to introduce today's speaker.
Awesome. Thank you so much, Lewis and Bethany, for getting us all up to speed on how the lecture series will work. So today I have the great honor of introducing Dr. Rafaela Missagia. Rafaela is an evolutionary biologist and assistant professor in the Department of Biology and Institute of Biosciences at the University of Sao Paulo in Brazil. Rafaela's research focuses on the morphological diversity of mammals and its correlation with ecological aspects, in particular investigating the adaptive aspects of predatory behavior in rodents. Rafaela is also dedicated to promoting gender diversity in science and is a founder of the Women in Science group at the Federal University of Minas Gerais, where she received her master's and PhD degrees. So today we're very excited to hear from Dr. Rafaela Missagia on advancing morphological research with computed tomography, a case study on predatory rodents. So please join me in welcoming Rafaela. Take it away.
Thank you, Erin. I'm very happy to be here today as part of the Palaeoverse Lecture Series. This talk is not exactly on the paleontology team, but several of the ecological inferences that I will discuss here based on morphology are applied to paleontological or fossil material. So you all may have noticed that I mentioned predatory rodents in the title of this talk, which may not be very familiar to some people or to most people. That's because rodents are usually known as small and opportunistic animals. And part of that opportunistic behavior is related to their skull morphology. They're well-developed, ever-growing incisors that are separated from the cheek teeth by a long diastema, allows access to a great range of resources compared to other mammals and other vertebrates, including mechanically resistant resources that they can gnaw and reduce, such as hard nuts and seeds. And when we think about rodents, we see them more as prey than predators. Usually when we think about predators, we think about mammals of the order Carnivora, which includes, for example, big cats and animals like wolves, and basically a big, ferocious, and bloodthirsty hunter. We usually don't think about a mouse, but if you look at this little guy, it looks pretty much like a predator. It's eating another mouse. And this is Onychomys, one of the most famous predatory rodents, also called the werewolf mouse and the howling mouse, because of this. I hope you guys can hear the sound of the video. I don't know. Well, I don't know if you guys are hearing, but basically showing the mouse howling to the moon as a territorial behavior. So Onychomys, as I just said, howls to the moon, which is a more familiar trait of predators that we are more used to. And Onychomys is a very aggressive hunter that feeds on scorpions and centipedes and grasshoppers, and even small vertebrates like lizards and other mice that can sometimes be even bigger than themselves. And as you can see in the video, they can hunt in packs. And these peculiar features have made this species very famous, this genus actually. Quick search shows several headlines that describe Onychomys as a badass and hardcore mouse. And Onychomys may be the most famous predatory rodent, but it's not the only one. During the evolution of rodents, some species became very specialized predators. While many rodents can occasionally eat other animals, we're talking here about species that underwent significant changes in their phenotypes in response to the demands of capturing and consuming live prey, which are the ones we're calling here predatory rodents. We have, for example, different guilds of predatory rodents, like semi-aquatic fish-eating species that we can see here in the left, in the picture, in contrast to shrew-like earthworm eaters, as we can see in the right. And each of those have evolved independently in different lineages of rodents and multiple times during rodent evolution. And the species show a wide range of adaptations. Some of them appear to have invaded these new niches, like the semi-aquatic and fossorial niches, where they could probably find prey more easily, adopting different life modes, and developing some locomotor adaptations like webbed feet for swimming and developed claws for digging and even for finding their prey. And Onychomys have evolved even resistance to prey toxins through modifications of some metabolic pathways. And this is a great example of the wide range of adaptations that these species show. And in Onychomys, instead of inducing pain, the sensory receptors can bind to the toxins and use them to block the pain signals. So we see here like a good example of physiological adaptation to the predatory behavior. And besides the aggressive hunting behavior of Onychomys, we see some other behavioral specializations. In Hydromys, for example, which is a semi-aquatic predator that hunts invasive cane toads in Australia. It was documented that they show a preference for larger toads when comparing the size of frogs or toads that fall into traps. And this species, Hydromys, is able to find and consume specific non-toxic organs only, like the heart and liver. And in some cases, Hydromys even removes the entire venom gland before eating other parts of the animal. And of course, we cannot talk about adaptations related to diet without mentioning the gastrointestinal tract. So some of the species eat chitinous exoskeletons of invertebrates, and this could damage their tissue, their stomach tissue. So some of the species have some protective pouches to that house this glandular tissue of the stomach to not be harmed by the material they ingest. And of course, we also have to talk, when we talk about diet specialization or trophic specialization, about the morphology of the masticatory complex. So I always like to show this example, which I consider the most extreme example of morphological adaptation for trophic specialization, which is Palaeoverse vermidax. It's a rodent species that was described and found only in 2012, and is the only species of rodents that does not have any molars, which, as you may know, or some of you may know, it's a very particular characteristic of rodents, their ability to chew and gnaw. And the species simply lose the molars. And in addition to the absence of molars, which indicates that they have a very soft diet and do not chew much, Palaeoverse also show an elongation of the rostrum, as we can see here in the red circles in the figure, when we compare with generalist species. And we will explore the connection of the elongation of the rostrum and diets later in the talk. So some years ago, for my PhD, I decided to investigate the morphological evolution of a group of neotropical rodents called the Akodontines, or the Akodontini tribe. And the Akodontines are part of the Sigmodontine radiation in South America, or the neotropics. And they are the second most diverse in number of species, with about 90 species recognized today. So here we have the Akodontines. We have, of course, some more generalist species, but also more specialized, fossorial, semi-aquatic, and predatory species. And they are quite diverse in morphology, as we can see in the pictures, and not only in external morphology, but also in school shapes. And this is why I select them as the focus of my PhD research. I wanted to know if their diversity of school morphologies could relate to their different diets, focusing on species that feed on other animals or predatory species. In order to do this, we decided to explore the functional morphology of their jaw by quantifying the mechanical advantage of their masticatory muscles. So mechanical advantage is a proxy that describes a trade-off between velocity and force, which means that the jaw can be either strong or fast. Our main premise here was that faster jaws, and hence lower mechanical advantages, would be found on predators that need to capture live prey. And this would lead to weaker bites, telling us that the species were giving up some more resistant resources, as seeds and grass, to rely mainly on soft animal meat. So we calculated the mechanical advantage of jaw closing muscles using each muscle's lines of action combined with the distance from the biting point to the jaw joint in the skull. And we also estimated the bite force using incisor variables related to bending resistance. So here we have the jaw-shaped morphospace plotted over heat maps of bite force in A, and mechanical advantages of three jaw closing muscles in B, C, and D, showing that the predatory akodonts depicted in the figures in triangles have in general lower bite forces and lower mechanical advantages in general, of course, which characterizes a fast and weak bite. And what we see here is a phylogeny of this species plotted over the mechanical advantage morphospace. And we can see that predatory species in blue invaded this morphospace area in the left more than once, representing what we can call a functional convergence, with more slender and elongated jaws that have in general a faster gait and weaker bites. And this also shows us how specialization, in this case trophic specialization, can increase the morphological diversity of one group. But this is only one evidence of adaptation, since the basic patterns of jaw movement associated with feeding or with diet have more than just one component. We should consider not only the shape and angle of the jaw, as we kind of just did, but also teeth morphology, teeth morphology, and of course the anatomy of muscles, or masticatory muscles in this case. So to dig deeper into this, we decided to take a closer look at the muscle morphology of acondyles. We wanted to compare the masticatory muscles of predators, represented here by two species in the red square, Oxymicturus delato and Bladenodes breviceps, with omnivore species or non-predatory species, represented here in the blue square by Necromis and Codon. So basically, we wanted to know if the functional adaptations that we saw on the jaw were also reflected on muscle morphology, since the jaw is basically area for muscle attachment. And in order to investigate these morphological complexes and other ones, as I'm going to show you, we began to apply microcomputed tomography, or CT scans, which is an imaging technique that uses X-rays to create high-resolution 3D images or models of objects, in this case, mouse skulls. And this technique is especially useful for us to access structures that are challenging to study with traditional anatomical methods, either because it provides a more clear picture of these structures, which means that we can see them more clearly, or because it does so without any damage to the specimen, which is always desirable. And more specifically, for the muscle morphology, we chose to use the DICE-CT technique, also called diffusible iodine-based contrast enhanced computed tomography. And this is a long name for, it basically describes a technique that uses a solution that binds to soft tissues. In this case, the iodine solutions binds to soft tissues as muscles to make them visible in X-ray images. As you all may know, X-ray images shows dense material like bones. And for us to be able to see more soft material, we need to use some contrast, in this case, iodine. And DICT represents what we can call a novel means to recover in situ internal anatomy, because it gives us anatomical context and has been heavily used in the last few years to explore basically every morphological complex of vertebrates and also invertebrates. And another advantage is that it's a non-destructive technique, as I mentioned, as all CT scans in general, which means that it can be done without permanent damage that occurs in traditional dissection techniques, because we lose the specimen and the material as we go through the dissection. So specimens can even be destained after scanning, which makes this technique reversible. And lastly, the resulting images or 3D models, in this case, can be shared widely with the scientific and the non-scientific community. And we even have some specific digital repositories dedicated entirely to this kind of data. So I'm going over more details at the demonstration part, but this is an example of what we have after the process of staining and scanning the specimen and segmenting each slice. And you can see in the upper corner, I slice. And I'm going to see this in the demonstration too. And we can see in the screen a 3D model, which is each muscle identified. And this is one of our predatory rats, Oxymicturus delato. And one of them, at least what I see as one of the most cool aspects of this technique, is that we can move this model around and make it big. So we can see some structures that are not so easily seen, even if you have the specimen in hand. And another advantage that I forgot to mention is that sometimes we don't need to have the specimen in hand. We can have the specimen scanned, even in other collections in other countries, and have the models sent over to us by some digital repository, for example. So it really allows access to more material for morphological studies. And we can break down all distinct layers of each muscle mass that we saw in the whole skull which is, of course, very useful when we're talking about anatomical and descriptive studies. In this example, we have, for example, the masseter, temporalis, pterygoid, and zygomaticomandibularis breakdown in each specific layer. And even better than that, we can compare the morphology of these muscles between species. So here's just a glance of our sampled species, the predatory and non-predatory ones. And let's see in more detail how these differences can relate to functional features that we discussed earlier, focusing on the masseter, that is the blue muscle mass, and the temporalis, which is the red muscle mass. So first, the temporalis, we saw that this muscle is larger in predators. And this muscle is not so active in generating bite force as the other ones, being more important to stabilize the jaw joint during biting. And the jaw joint is basically where the jaw connects to the skull. And this muscle is responsible to restrict lateral and anteroposterior movements of the jaw, which you may think can be very useful when we talk about predatory species, not only for predatory rodents, but also other predatory mammals that usually have enlarged temporalis muscles. And we can compare this to other species of predatory rodents besides the one we are analyzing. So some of them have been dissected, and a larger temporalis was also observed in Hydromis, that semi-aquatic Australian rat that eats canetons that I mentioned before, suggesting that jaw stabilization is important for other predatory rodents. And concerning the masseter, we saw that this muscle has a smaller volume on predatory species that we are comparing here. So the masseter is the muscle that is most involved in generating bite force in the masticatory complex. And we also see that it has a more anterior attachment areas in omnivores or non-predatory species, which basically increases its mechanical advantage and also its bite force, meaning that predatory species have lower bite forces. And also, together with small volume, comparing predatory and non-predatory species allows for a wider gape in predatory species, which is very useful when you're subduing prey. And this is in line with our previous findings of smaller areas of attachments for the masseter in the narrow zygomatic plates of predatory species, and also with our lower estimated bite force for predatory species. And a less developed masseter was with a more posterior area of attachment was also observed in other predatory rodents as onychomis, hydromis, and ichthyomis, which shows us the importance of a wide gape for predatory species to be able to catch their prey. But this later species that we are not analyzing here, the other predatory species, have greater mechanical advantages to compensate for the lower muscle volume in order to be able to capture and eat the prey. And this was not observed in predatory acodontines, since they present smaller mechanical advantages and smaller masseter volumes. And this may be related to differences in food items consumed by each of these predatory species. Onychomis, hydromis, and ichthyomis, for example, the ones that have higher mechanical advantages, include small vertebrates in their diets, which may have higher mechanical demands compared to the acodontines that eat mostly small invertebrates, which may mean that bite force is not that limiting for them. And the adaptations related to feeding mode seems to reflect not only predatory behavior, but also the mechanical demands of different types of prey, which shows us that we cannot always consider predatory species as just one guild. They have to be, we have to think about them in terms of what they are eating. Okay, so we talked about jaws and we talked about muscles, but what about dentition, which is a feature directly related to diet? So this project that is led by PhD student David Villalobos-Chaves and his advisor Shalin Santana, David collected CT scans of acodontine species, the ones we are talking about here, including predatory ones, and created these 3D models to calculate 3 dental topography metrics, which are quantitative methods for characterizing ecological or dietary groupings using teeth morphology. And these metrics include shapes and angles of cusps and surface complexity, for example. And so far, this is still an ongoing project. David observed some differences between diet specialists and generalists across this metric. For example, he found that herbivorous and predatory species, which are dietary specialists, have sharper molar occlusal surfaces compared to omnivores or generalists. So these sharper occlusal surfaces seems to provide an advantage for cutting through food items with specific material properties, which are more common to specialists. Omnivorous species, on the other hand, have more complex molars with larger occlusal surfaces. And besides the masticatory complex, predatory species have to be able to find their prey, which may involve some sensory adaptations. So we started investigating the brain of predatory species through their endocasts. And the endocast is what we see here in purple in the right, and is a model made from the internal cavity of the brain case, which we can also see here the picture in the left that shows a skull cut in half. And it is assumed that the endocast is a reliable reconstruction of the brain itself, at least in rodents, and is heavily used not only in fossil material, in which it would be possible to access the brain itself, but also in living specimens and species in which the brain was lost, the preservation process. And it's important to point out that endocasts can be made, and more commonly made, with resin or other plastic material, which can cause the integrity of the material, highlighting again the value of virtual endocasts and CT scans, which can be made without any damage to the specimen. And with endocasts, we can compare specific areas that are thought to be related to hunting activities, such as the olfactory bulbs. And the olfactory bulbs, highlighted here in blue, receives neural input about smells that are detected by cells in the nasal cavity. And we would expect larger bulbs in predatory species, considering that they can use the sense of smell to find prey. So we have this project led by master student Arthur França, in which we are investigating the size and shape of olfactory bulbs of Akodontine species, focusing on predatory species. And these results are also still preliminary, and we are in the process of understanding this data. But we saw so far that Larinois breviceps, one of our predatory species, that in this case has a small visual capacity, did show the widest and highest olfactory bulb. And the olfactory bulb in the species proportionally occupies the largest part of the total endocast volume amongst all analyzed species, which indicates for us probably a high olfactory capacity. And still in the same topic, we are also beginning to explore the turbinal bones of predatory rodents. So turbinals or turbinal bones are basically the bony plates covered by vascular tissue that projects from the internal walls of the nasal cavity, and they serve two primary functions: thermoregulation and olfaction. And it has already been seen that some predatory rodents, specifically those that eat earthworms, have a much more complex turbinal structure in the olfactory portion of the turbinals. As we can see in these figures by comparing these structures inside the red squares. And we see that predatory species have more complex olfactory turbinals in the sense of having larger surface areas compared to non-predatory species, which may also be related to a more developed olfactory sense, helping them to detect prey. However, neotropical species have not yet been truly investigated, and this is the goal of the project led by undergraduate student Fabrício Oliveira. We are beginning to investigate the morphology of the olfactory turbinals in apodontines in order to compare predatory and non-predatory species, as we have been doing for all other morphological complexes, and to try to understand whether predatory species also use olfactory turbinates to find their prey. So Fabrício started working on this just a few weeks ago, so we don't have even preliminary results yet, but we hope to have them soon. So with this, with all this investigation, putting together different morphological complexes, we intend to have a better understanding of this trophic specialization in neotropical species by integrating the investigation of different morphological complexes of the skull of predatory odontines, and integrating classical techniques like functional morphology, which I talked about in the beginning of this talk, with comparative anatomy based on 3D models, highlighting the importance of these new techniques for acquiring morphological data and comparing morphological data. And with that, I'd like to thank my collaborators and co-authors and curators and staff of Memo Collections that kindly gave access to specimens, funding agencies, the Palaeoverse Talk Series, and Harry Dillon for having me here today, and all of you for being here listening. I'll be happy to take any questions, but I believe it's better if you go to the demonstration part. A quick demonstration of the software, the 3D slicer software that we are using to access all this morphological data based on CT scans images. So just some details. We can do CT scans on, as I said, bone material, which is still more common, but more recently it has become more heavily used, the technique of the dye CT, where we can use some contrast to be able to see soft tissues, as I mentioned. So just to give some more details, we follow this protocol of Jamie Gray, in which we having the skulls with, of course, the masticatory muscles attached. These skulls were immersed in a solution of iodine after being, I don't know how to put this, but before the iodine, they had to be put in solutions of progressively less concentrated solutions with alcohol, because this material is more commonly stored in alcohol. So to take off some of the ethanol of the material before staining, we follow this protocol of leaving 2 days each in different concentrations of ethanol, and then they were put in the iodine solution for approximately 2 weeks, depending on the concentration of the solution. In our case, was 2 weeks because it was the lowest concentration. And then they were scanned in a SkyScan 1174 on the University of Minas Gerais. And then after scanning, we have, as I showed before, the slices or X-rays, as we may put, that together make the 3D model. And here you have an example of a slice, and we can identify the different muscle masses on each slice, as I'm going to show on the software. Okay. And So this is the 3D slicer software. It is open software, open coded software, which means that people can develop different modules and that can be integrated into the software. One very famous module for anatomical and functional morphology and comparative morphology studies is the slicer morph, and it has like different functions that help us to segment and to visualize this material. So here we have a specimen already open here in the 3D slicer. As we can see, we have 4 different screens showing different cuts of the material. So here, like a coronal cut, transversal cut, and a sagittal cut, which is very useful when we are trying to identify different masses or structures, because the structure may not be so easily seen in one view, but more easily seen in another view. So it really helps through the segmentation process. So for the ones that there are viewing the lecture and are so familiar with the terms, when I call what I call segmentation process is basically choosing or targeting one morphological, painting it over or highlighting it in the software for the software to understand the structure that you want to see. So it can be very manually. It depends on the condition of the scan, the quality of the images, but 3D Slicer has all these tools here on the far left of the screen that can help this process of segmentation by, for example, highlighting the parts of the tomography that have higher density, like bones, and you can understand that and highlight only this part, and it may function in some cases for muscles, because our main goal with the staining process is to make all heavy to be able to be treated by the scanning process. So we have like a big mass of very dense material, including muscles and bones. And in my case, I had to do manually. For example, I can, this is one of the views. And when you do this, as you can see, go over all the specimen, slice by slice, the whole skull, and you can see the muscle masses start to appear here. And at this point, we just have to, if we're lucky enough, like other studies, to guide us through this process. I did. Depending on the structure, maybe a little more difficult, but it's basically trying to see the structures you want to segment, in this case, the masticatory muscles. And it's basically what I did. So here we have the module called the segment editor. As you can see here, I did some muscles segmented. So for example, the pterygoid, and you have basically to paint over each slice. And as I said, we have a lot of tools to make this process more easy to do. Like, for example, you can do, you don't have to do each slice. You can do one slice, skip 5, do another slice, and ask for the software to interpolate this, and he will understand how to, or the better way to fill the empty spaces. So that can save some time, for example. And just so you see, like if you want to segment another muscle, we just can add another segment, as you can see here, segment 3, and use one of the tools. The simplest one would be painting, and you're gonna just paint it over to tell the software that this part here is what I want to see in the final 3D model. So as I said, you can go like slice by slice, or just jump some slices do another one. Of course, trying to be very much like more as detailed as you can. You can really go over the pixels and do again more detail as you can. So I have some segments here. The muscle that we saw, for example, and go back here, and even the brain already here. Let me see if I can show you. You need like let's say very efficient computer to run these programs and do this analysis. They take a while. So here we have like the 3D version of this already segmented muscle masses. And another very cool aspect that we saw on the results that are presented is that this software can very easily calculate the volumes and surface areas of each muscle mass in this case, or of each structure that you are interested in segmenting, and this can be compared or made like proportions with the whole volume, and you have some quantitative information about your study object. Okay, I think this is it. I'll be happy to take questions if you have any. Thank you.
Awesome, wonderful. Thank you so much for an excellent talk. I think at this point we will take questions if the audience has any, and I will also stop the recording.